Good evening, everyone, and welcome to National Science Week, as well as our first sci-fi series event of 2022, posing the question, is RNA the answer to cancer? I'm Tegan Taylor, a health and science reporter for the ABC, and I am very delighted to be hosting this evening's discussion. RNA is at the core of our biology. It's the software of life. It translates our DNA into the very products and processes that make us human. As well as being the key ingredient in some COVID-19 vaccines, RNA technology might become the key to treating conditions like cystic fibrosis and muscular dystrophy. The next steps for RNA as a therapeutic lie at the interface of biology and medicine, chemistry and technology. We're going to dig into this tonight with some leading experts in the field, who I will introduce to you very shortly. But before we begin, I'd like to acknowledge the Bidjigal people that are the traditional owners of this land. I would also like to pay my respects to the elders, both past and present, and extend that respect to other Aboriginal and Torres Strait Islanders who are here today. And just to note that we're recording this event. I don't know why you would possibly want to leave any earlier than the full thing, but if you do need to leave early, the link to the recording will be emailed out to everyone who registered in the coming days. The last few years have been challenging, to say the least, but emerging from our experiences with COVID-19 has come an area of research that has burst into the zeitgeist, RNA. But its applications extend far beyond vaccines, and there are a few people more qualified to explore this topic than our panellists today. Up first, Professor Palli Thordarson is Director of the University of New South Wales RNA Institute and President-elect of the Royal Australian Chemical Institute. Welcome, Palli.
Thank you, Tegan.
And Conjoint Professor Maria Kavallaris is Co-founding Director of the Australian Centre for NanoMedicine at the University of New South Wales and Head of Translational Cancer NanoMedicine theme at the Children's Cancer Institute. Welcome, Maria.
Thank you, Tegan.
Unfortunately, Conjoint Associate Professor Joshua McCarroll was unable to join us this evening. His insights and expertise will be missed. So, Palli, let's start with the basics. What is RNA?
Well, as a chemist, RNA is one of these wonderful molecules that we find in biology, and we're all made out of chemicals. It's one of the three main classes of what we call biomacromolecules. We got DNA, and one way of thinking about DNA is that's like the hardware. That's where the information is stored inside every cell of our bodies. And then we got RNA, which is really the software that takes that information and transmits it around the cell. And one of the output of that process are the proteins that do a lot of the work within our cells and communication to the external world. What we always thought was that, or maybe thought for a while, that RNA was just sort of a bit of a messenger, which is where the M in mRNA comes from. And that is certainly one of its major roles. It is a messenger between the DNA and the protein, but it also has all the roles within the cell in terms of organizing the cell and even in the structure of the cell. So it's one of the three key biomolecules.
What's the evolutionary purpose for it? Why do we have it in us?
Well, I think, again, if we think about that software analogy, that's maybe where it comes from. But going way back, and we were just talking about this earlier before we met here. I'm a chemist. My PhD was on chemistry that relates to trying to understand how life originated on this planet. And we think the first molecule was a molecule that could do both information transfer and do work. Now, RNA is also very unique that way. It's actually the only molecule that can do both. So that's kind of why it sits at the center of everything in life. It was probably the first, it was the key building block from that prebiotic soup of very small chemicals that were not doing anything interesting to what we recognize as life. It was the magical molecule.
I think one of the other things, Palli, is also unique about RNA in the cells is that it also regulates itself. So there's all different forms of RNA, and some of them regulate the gene expression, or they can suppress gene expression. Make it not express certain genes and proteins, etc. So I think it's unique in that way that it's self-regulated and it regulates every aspect of the cell through that.
So we're going to have a look into different forms of RNA tonight, but we've actually got a little video to show you to explain the type of RNA that we're going to dig in most on tonight, siRNA, and the applications it might have for cancer. So let's take a look at the video now. mRNA is well known as the key ingredient in COVID-19 vaccines. But the technology could also play an important role in how we treat cancer in the future. siRNA, known as short interfering RNA, are small sequences of genetic code that can interfere with genes that are driving cancer growth and survival. Scientists can synthetically engineer these genetic disruptors or blockers in the lab with a specific siRNA sequence. These siRNA can be delivered into the body and target cancerous cells. The siRNA navigates around the body in a protective oily capsule until it can enter the tumour site. There, it delivers the siRNA sequences that match the cancer genes, knocking them down and causing the cancer cells to die. UNSW researchers are continuing to advance the development of the technique, including how it might combine with other drugs to help in the fight against hard-to-treat cancers. What a great voice that voiceover person had. Okay, so Maria, talk us through the different types of RNA and why siRNA is so important.
Sure. Look, I'll probably start with the problem. And one of the areas we're working on is on cancer and difficult to treat cancers. And one of the challenges is that we know a lot now, and we're learning more and more about the genetic makeup of cancers. We're understanding that there's particular genetic mutations or alterations that are happening, but we don't actually have drugs for all these alterations. So one way to tackle some of these cancers is to deliver RNA type technology to the cancer cells to try and knock down genes. So one of the areas we're doing a lot of work in my lab and Associate Professor Josh McCarroll's lab is to actually use what we call short interfering RNA. So you saw that video. These are very short sequences that we can actually design to target very specific cancer genes. And so often if a gene, there's too much of it in the cell, it helps drive the cell. It's like putting fuel into the cell to grow and divide and get out of control. So if we can actually put a spanner in the works, which is the siRNA, and block those genes, then the idea is to stop the growth of the cancer cells. So that's one application for short interfering RNA technology in cancer. But there are other forms of RNA technologies as well that I can expand on.
Yeah, let's talk about it.
All right, so another area that's gaining a lot of momentum is in the area of, I'll go back just a little bit. So when I think about RNA technologies and what they can do for cancer, I think of things like therapeutics, like I just described. I think of diagnostics. So we can also use the sequences that are released in the blood from tumour cells to track the emergence of cancer, but also how a patient's responding to therapy. So that's what we call liquid biopsies and doing things like that. And I also think about things like cancer vaccines. So where we can develop, and this is a very big area of focus around the world, to develop vaccines against certain types of cancers. So we know, for example, we know about the cervical cancer vaccine, which has been a huge success, but now there's a lot of effort around mRNA vaccines, not dissimilar to, I guess, what we saw with the COVID vaccine, but targeting specific types of cancers. So that's a really exciting area as well. It's
pretty wild that there's so many applications that you've discovered when RNA is actually relatively recently discovered.
Not really. I mean, it's recently come into the spotlight for the public. mRNA itself was discovered in the early 60s. And in fact, even what we now recognize as mRNA vaccine, people were doing experiments as early in around the 80s that sort of established the principles on which mRNA technology is now used in the vaccine space. The siRNA were discovered in the late 90s, if I remember correctly.
Yeah, 98 and won the Nobel Prize.
Yeah, pretty soon after that. So it's not completely that we just found this yesterday. And actually with the mRNA vaccines, one reason why they could develop them this quickly when COVID hit us was actually all that work that already happened, both in the siRNA and the mRNA space the last 10 years. And a lot of that work was actually targeted towards cancer therapies. And in fact, Moderna, for instance, was initially established to develop cancer therapeutics, and they still work in that space too.
Yeah, I think there's been enormous growth, as Palli said, we've known about mRNA for a long time. I think over the last 20 years, though, there's been an explosion, and probably even less than that, an explosion of information about these different types of RNAs, like, I don't want to rattle them all off, but there's things like long non-coding RNAs, there's short interfering RNAs, there's circular RNAs, and they all have different functions in the cells. But also, the more we've understood about those, the more we've realised these are potential therapeutics in the future for different types of diseases, including cancer. So there's an enormous effort to better understand how these things are regulated in the cell, but how can we also exploit them for therapeutic applications?
Has the publicity around the mRNA vaccines and COVID helped people understand more about what you do, or has it maybe fueled misunderstanding? They think that all we're doing is vaccines?
No, I think it's mostly helped. I have done quite a lot of work on trying to explain to people that mRNA vaccines are just the tip of the iceberg, right? A very important tip of the iceberg, to be said, but there are other aspects of RNA, both in terms of the actual chemical makeup, the difference between siRNA and mRNA is simply mainly the length of the molecule, and then the therapeutics that we can use for them. So and I think people do understand that as soon as I pointed out to them. This is just a starting point. Like, I know the public only saw RNA therapeutics first in the form of mRNA vaccines 2 years ago, but as Maria said, we've been working on this for a couple of decades across the world.
But I think one of the advantages in terms of these vaccines is I think the general public, know they may not fully understand all the aspects about vaccines, but it's put mRNA on the map, really.
Yeah, I think general health literacy in the general public's gone up a lot, especially around genetic sort of things. But let's talk about cancer specifically. That's what we promised people that we would talk about. Maria, in terms of cancer therapeutics, let's start there. What are you exploring and sort of where are you up to with that research?
Yeah, sure. Look, one of the I should say I predominantly work in children's cancer. I also do some adult cancer, but that's my primary focus. And children's cancer is the most common cause of disease-related death in children in Australia. And so one of the successes in cancer therapy has been children's cancer, because there's been more advances in children's cancer than any other sort of area. The problem is it is still the number one cause of disease-related death. And what this means is that the patients, the 30% of patients that we identified have the worst prognosis are often given very high-dose chemotherapy. And so they get very sick. Many of them will be in hospital with the side effects of the very treatments that are meant to save their lives. And one of our passions at the Institute and in my group is to try and develop more effective, more targeted and less toxic therapies for the children. So hence where our interests came. And we started off where we are now, which is with siRNA, which is the short interfering RNA, to try and deliver this material to the tumour cells and stop the growth of the cancer cells. So we pick genes that we know are involved in either controlling cell division and multiplying and spreading, and also that are very, very sort of aggressive genes for the growth of the cancer. So by really interfering with that, trying to deliver the material to those tumour cells and blocking the growth. And then sometimes we also work on aspects to combine that with some conventional chemotherapy. So we're looking at ways of actually also reducing conventional chemotherapy, but still getting very good efficacy to knock down the tumours. So some of the cancers that we work on are some of the most aggressive cancers. So we work on some of the pediatric brain cancers. And unfortunately, in terms of the cancers, brain cancers are the number one cause of cancer-related death in children. And we also work on some of the aggressive leukemias. Leukemia is the most common cancer children get. we work on cancers such as neuroblastoma, which is 70% of the children at diagnosis, the cancer's already spread around their body. So if we're developing ways to try and deliver this sort of sRNA material to the tumour cells, try and get it knocked down and destroy the tumour growth.
Now, I should have said at the beginning, folks, that you can actually ask questions tonight. We want to hear from you, and you can do that by going to Slido, and the hashtag is RNA Cancer, hashtag RNA Cancer on Slido. And we've actually already got a question that kind of speaks to what you were saying, Maria. Someone saying, do you think RNA will take over chemotherapy treatments in the future?
I don't think any of these treatments will take over completely, but what I think is they'll be highly complementary. So I think what we'll be seeing is that, just so you know, in terms of cancer therapy, monotherapy, as in, just one single treatment rarely works for any, whether it's children or adult cancer. It's combination therapies that have been the magic in terms of improving survival for patients with cancer. So again, I'd be thinking that what we'll be doing this is as a complementary therapy with different types of therapy. So we're starting to already identify where we can knock down a particular gene and sensitize to a whole range of drugs. And combining these, I think, is what's going to be the magic.
But you were saying before that these are often rare conditions. Is the benefit of working with something like RNA that you can modify that genetic code to sort of tailor it to whatever's going on?
Yeah, and I would add somewhat a chemistry perspective here. In medicinal chemistry, people sometimes talk about structural therapeutics, which is most common medical drugs are based on. So you make a molecule has a certain structure that has a certain function, binds to a receptor, whatever. RNA is quite unique in a sense. It's really what we would call an informational drug. It's the information coded. Coming back to the software analogy, we make a drug that has information coded in it, and then that can target a rare type of cancer where, in combination probably still with other things, right, Maria?
Yeah.
But if we can target that particular gene that is overexpressed or whatever, that with an informational drug. Right?
And you made a good point there, Palli. The other thing is also being able to target multiple genes at once.
Yep.
So you don't necessarily do it. When I talk about cancer's complex disease, so I should say it's not a single disease. There's over 200 cancers, and there's even within the same patient, tumours growing in different parts of the body may have different genetic mutations that are driving the growth of that cancer. So that's why cancer is so complex, because sometimes, you'll treat the doctors will treat the patient, the tumour will shrink, the patient seems to be doing well, and all of a sudden, these other tumours pop up in different sites. And often they were cells probably from the original tumour, but they had genetic alterations that made them resistant to therapy. So the more we're understanding now about the genetic makeup of these cells means we're hoping in the future we can rapidly adapt therapy. So I'll use the analogy of the COVID vaccines, where we know we've got all these variants of COVID now, and now they're more rapidly moving to put new sequences within that delivery vehicle, which is the nanoparticles that go that deliver the material. We can do, in the future, I'm hoping we'll do the same with cancer. And teams like Palli's who can actually then synthesize these things for us rapidly, and hopefully we'll get to a manufacturing situation where we can then adjust the treatments according to how that patient is going.
Can we talk about how you actually get that little piece of genetic material into where it needs to go? You can't write an address on it and just sort of post it into a patient. How do you make sure it gets to where it needs to go?
Well, we have different ways. So sometimes, so let's just say we're looking at a liver cancer. Remember the little video that was shown earlier?
Yeah.
It showed how there was like a liquid or oily layer where the material was in the centre, and we call those lipid nanoparticles. So the lipids are sort of the fatty emulsion, if you want to call it that. Now, when they circulate around the body, they often do go to the liver. So if you've got liver cancer, that's actually a good thing. But many cancers are in other parts of the body. So research that's going on with people who are at the UNSW RNA Institute and also the Australian Centre for NanoMedicine that I co-direct, we're working on ways to make them go to different parts of the body. So one way you can do that is you can attach an antibody to the surface, and that antibody not only recognises that nanoparticle with the RNA, so imagine, but the surface of that antibody recognises the cancer cell. So if you have a particular cancer that it's got what we call a receptor or a little flag on it, as the nanoparticle is circulating, if it comes in close proximity to that flag, it will attach and get taken up by the cancer cell. And we've actually found that we're improving the uptake of these nanoparticles into cancer cells using these antibodies. But you can use things like peptides, like Palli, this group has been developing as well, that we've been working on to also attach those onto the nanoparticles, which contain the genetic material in them, the RNA, to get to the tumour cells.
So that's the structural chemistry that you're talking about.
It is, but it's also what you said, actually, in a sense. We are trying to put addresses on the particles of the cargo. You're absolutely right, whether it's through antibodies or with peptides or other, what we would call ligands. Yeah. And it is honestly one of the key challenges in the field, but it's also partly what we set out the UNSW RNA Institute for. We think this is the most important challenge we can help people like Maria and others to try to get to the final target, which is to develop new therapeutics. It's also actually saying a broader level, it is one of the biggest strengths that Australia has in this field. We're really strong in this field when it comes to designing better delivery systems.
Yeah, it's where biologists and chemists and engineers and physicists and everyone has to talk to each other and learn to speak the same language, which is not easy, let me tell you. But it's where the magic happens, really, is when we can all team up in this way to really advance the field.
Right, so what level are we at? Like, what stage of research are we at? Is it dishes? Is it animals? Is it humans?
At the moment, look, within the RNA Institute and the work we're doing, it's more, we make, well, we've got two levels. There's the sort of, you call it the dishes. We're also, we can make mini tumours in a dish now, so we can create little mini tumours. We use bioprinting to do that. We can rapidly screen our nanoparticles using that approach, and then once we know we have something useful, we then would have to go to the animal studies, but it helps us minimise the use of animals by doing that.
Just going to the Slido questions again, there's a question about cancer vaccines that I might get back to, but there is someone else asking, will this treatment method come under personalised treatment? So can we talk about how you're tailoring to a particular patient, like you said before?
Well, that's exactly what it is. What I've been discussing here is personalised therapy. So from identifying what surface receptors are on a patient's cancer cells, so you can tailor the delivery, to identifying what genetic mutations are happening. So at the Children's Cancer Institute, where I also work, there we have the Zero Childhood Cancer Program, which is a personalised medicine program, which is Australia-wide. So we get all the samples from patients around Australia that come to the Institute, and they're sequencing the genetic material from the patients, and again that gives us opportunities to start tailoring for some of these really challenging cancers specific treatments. So it's tailoring both in terms of recognising the cancer cells, but also what's going wrong in the cancer cell and trying to target that treatment.
And for personalised medicine, RNA is probably the best choice, because it actually comes down to the manufacturing too, because as soon as Maria and her people can work out what does a gene has to be targeted, what does the RNA look like, it is a little bit like you said earlier with the current COVID-19 vaccines, because it's an informational type molecule, it's just like software. It's relatively easy to make that slightly different siRNA that targets this particular cancer that is in this particular person with that turnaround. I mean, this is different from an infectious disease, but for personalized medicine to really work, we want to have the drug ready within a few weeks of diagnostics, right? And that's going to be very hard with any other type of manufacturing methods, but RNA has that ability.
What's the rest of the manufacturing process look like then? Like, what's the base model that you're plugging this into?
So there are actually mRNA vaccines or mRNA versus siRNA made in very different ways, although they're both informational molecules. For mRNA, what we do now, we actually create first a DNA template, and then you basically photocopy that 100 times with an enzyme, and you make a bunch of mRNA that way. And that's a relatively straightforward process then to adjust, to change slightly the letters inside, if you think about it like a string of letters. For the siRNA, it's a chemical process where we put one letter at a time on a string. It's a bit like you're threading pearls on a string. So, and that's actually also fairly straightforward to, like, you put in, 2 blue ones and then a red one and then a yellow one. And then if Maria tells me, no, this patient needs a green one there, we'll just change the yellow to green, and off you go.
How scalable is it?
It's quite scalable, and it's good you pointed out that part, and that's sort of what we're working on here, both in New South Wales and elsewhere in Australia, is to establish the manufacturing facilities to do this at the scale and quality required for human therapeutics. But RNA therapeutics are, in many ways, and again, looking at this as a chemist, more scalable than many other therapeutics. The sizes of the reactors and facilities required is actually quite small compared to, say, bioreactors that you need to use to make proteins, just to take one example.
Mm-hmm. There's a question about, can RNA-loaded nanoparticles be administered orally? And so I guess, brought it to that, like, what are the different ways that you actually administer these drugs?
There's people are working on that in terms of trying to deliver oral delivery. It just requires a different nano formulation for oral delivery, because obviously when you go orally, it's got to go through a very acidic environment in your stomach and then the gut. But people are working on nanoparticles, probably different to the lipid nanoparticles in the vaccines, but they're different types of particles. But people are working on this type of area as well. So I think in the future there will also be oral formulations available.
You mentioned side effects before when you're talking about childhood cancers. At the moment, the way they're treated is sort of sometimes just chucking everything you've got at it. What are the side effects that you're seeing with this different approach?
Well, because it's not in the clinic yet, so I can't tell you the side effects, but based on the sRNA therapeutics that have been approved for these rare liver diseases, and for one of them it's in children, they've been very what we call tolerable side effects. There haven't been any dramatic side effects. Even when they were doing the clinical trials, they never had to suspend the clinical trials. So that's actually incredibly encouraging, that there weren't. So I do not anticipate that there will be, but again, like any formulation, it would have to go through rigorous testing, and there's very high standards about clinical testing before something gets approved in the clinic.
So can we talk about some of the other applications you mentioned before? You were talking about cancer vaccines, liquid biopsies. Starting with vaccines, we know that the cervical cancer vaccine actually targets a virus that triggers the cancer later on. What are the other sort of cancers that you're targeting with vaccines or looking to target?
Yeah, well, see, we're not working on it specifically at the moment ourselves, but I do want to talk about it because I think it's important, because it is an area that is increasing in importance. So people are targeting vaccines that are actually looking at certain types of receptors on cells as well. So they attack certain types of cells. So I know there's work around the world around, for lung cancer, some of the more common adult cancers in terms of vaccines. It's going to be difficult to vaccinate against everything, and what you want to go after is ones viral caused cancers are the ones. We know that head and neck cancer has a strong viral origin as well, and it's not the similar virus that causes cervical cancer. Virus is related to that as well. So I think there's just interest in trying to vaccinate against some of the more common cancers.
But it's a bit like the COVID-19 vaccine, which is, one way of looking at vaccines in general, whether mRNA or not, you're trying to train the immune system to recognize something is wrong. And I guess that's the whole idea with the cancer vaccine.
Exactly.
Train them to recognize a lung cancer or
yeah.
Working out that biology is probably the tricky part, isn't it?
Yes, exactly. And you would have heard about probably CAR T cell therapy. And so people are now, using mRNA sequences to develop these CAR T cell therapies.
For our audience
well.
and maybe people here who aren't scientists like me, can you remind us what CAR T cell therapy actually is?
Well, what it is where you, at the moment, you can actually train the immune cells to recognise tumour cells. So, and it's been particularly effective for relapsed acute lymphoblastic leukemia, which is a blood cancer, where you can modify. Basically, they take the T cells out of the body, they put in a genetic construct within these cells to express what we call the CAR T cell receptor. So it actually recognises the tumour cells. That's probably the most simplistic way of doing it, to try and destroy the tumour cells. And it's now a clinically approved therapy, but it's quite expensive and it requires a lot of it requires a lot of work to do, and it has side effects. So people are now looking at using this mRNA technology to make the CAR T cells more rapidly and safer as well. But it is experimental at the moment. But that is a new type of cancer therapy that a lot of people are trying to exploit.
So a reminder, you can ask your questions by using Slido. The hashtag is RNA Cancer. There is someone asking, will cancer vaccines also require regular booster shots?
That is a good question.
It's a good research question. You should come and do your PhD at UNSW.
Yes, come and do that. I think it's going to require more work for that. So, look, I can't, and because I'm not a virologist, I can't really answer it. But yes, I would leave that to the virologists. But
Another question for you, Palli. Someone's asking about what's the cost of these? If RNA is the answer to cancer, what is the cost going to look like for these sorts of treatments?
I think in the long run, it is going to be moderate. Like any other new technology, what happens in reality is that those who put in the effort to develop it, they do need to try to get that investment back. So like any other new therapy, it's probably going to be a little bit expensive initially. But I have full faith in this technology becoming one of the more affordable cancer therapeutics. And as Mary said, I don't think we will have magic bullets based on RNA and we can stop doing everything else in the oncology field. But I think it will be one of the tools in that field that will be moderately expensive, because the manufacturing methods are relatively scalable and cheap compared to, say, CAR T therapy is a very good example. CAR T therapy is very powerful, but extremely expensive at the moment.
Because it's what we call a cell-based therapy.
Yeah.
They have to take the cells out of the patient. They have to modify them in the laboratory. They've got to expand them and then put them back into the patient. So, yes, it's quite an involved process.
Whereas when you think about it, the manufacturing cost of an mRNA vaccine is in the order of a few dollars per dose, right?
But you're making one thing for everyone in the country. You're making millions and millions.
No, that's what I mean. Like the economics are slightly different when you're doing cancer therapeutics, but the inherent manufacturing cost is lower than, say, with cell-based therapies.
And I think the more mRNA technologies or RNA technologies that are being used for different applications, because you can also see applications here beyond cancer for animal husbandry and things like that. The more it's used, the cheaper it gets. So if you think the human genome cost over $1 billion to sequence the first human genome, and now for $500 you can sequence the human genome. So the more you use things, the more things are made up, scaled up, the cheaper it gets.
What does that look like in Australia? What sort of industry might sit around this in, say, 20 years' time?
I think it will be a bit like our rest of our biotech industry. We already have in GMP, which is good manufacturing practice, which is what you need to make therapeutics to put into people. We already have in the protein space, we've got 3 or 4 facilities around the country. I think we'll have that in the RNA space within a decade or two. I mean, we already have at least 2, if not 3, in the works. So we've got the Moderna facility in Melbourne, which is for mRNA vaccines, and Moderna is working on, obviously. We got the New South Wales facility that will be built in North Sydney, which is more generic and agnostic to products. And We have Bioseen and Adelaide also has made public their intent to stand up some RNA production. So I wouldn't be surprised we'll have half a dozen fairly soon. And I think there's room for it because there's so many different applications and so many different types of therapeutics and RNA type materials that can be developed.
Well, let's talk a bit about applications. Before we get to, we want to get through as many questions as possible tonight, which we will. Again, go to Slido, hashtag RNA Cancer is the hashtag. But Maria, can you talk a bit more about the diagnostic side of things that you were saying before? I mean, that's where you save money, right? Like by getting in early and identifying something early.
No, that's right. I mean, you've got things like early diagnosis and also monitoring disease, like treatment progress as well. So this is where you can take small amounts of blood and isolate the genetic material out of the blood. So tumour cells themselves, our blood's always got sort of bits of genetic garbage going around. It's probably the best way to describe it. It's floating around. But because cancer cells have got unique genetic makeup, when they're releasing I'll call it part of its garbage, some of its regulatory systems, but they release it into the blood, then you can try and monitor the specific sequences of the genetic material in that blood. And we call that liquid biopsy. And it's particularly useful because you don't always have markers for different cancers, but it's particularly useful, say, if you know a particular patient has a particular genetic sequence that is altered in their tumour, and you can pick it up in the blood. Then as you might do surgery on that patient, and you start chemotherapy or any other type of therapy, and then you can take serial samples of the blood, because it's easy to take small amounts of the blood and monitor for this genetic material. And often there's different, remember we said at the beginning, there's different types of RNA. We talked about the short interfering RNA. There's also something called microRNA, which you find in the blood as well. And that can be, there's certain microRNAs that are markers of different types of cancers. So you can say, all right, the markers, you're monitoring, the marker is low, 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 and then suddenly the patient's off treatment, but the marker goes up. You can start investigating and saying, hey, the tumour may have come back. The doctors then have a cue there to start looking again to see whether something's happened, and then start adjusting therapy as well.
So it's less invasive than doing biopsies, but then also I suppose you don't have the same kind of manufacturing hurdles as we were talking about before, where you're developing a therapeutic. You just need to have the technology to read it.
Yeah,
is that right?
That's right. So we have highly sensitive, the polymerase chain reaction, which I'm sure the majority of
everyone knows what a PCR is now.
Yes, they all know what it's.
No
PCR.
one knew what it was 2.5 years ago.
After COVID, everyone knows what a PCR is. I don't know if they know what all the letters stand for, but they know what PCR is. Well, we use that technology, which again won a Nobel Prize when it was discovered. It's an amazing technology that allows you to say you've got a million sequences in a tube, and one of those sequences has a genetic alteration. The assay lets you put it through cycles of amplifying out that signal, so you can identify when there's a particular genetic alteration. So we use the PCR assay because it's highly sensitive to pick up these changes.
But I should add that although PCR is fantastic, it does have some limitations sometimes, especially if it's mixed in with blood and you're looking for low concentrations of your RNA. And we have people here within the RNA Institute working on the next generation of RNA sensing technologies that are even better than PCR. And I think they will be
nanosensors.
They will be very useful in, as you say, in oncology.
Exactly. Well, yes, we published a paper with Justin Gooding a few years ago, is exactly on that, taking blood and diluting it just 1 in 2, and directly being able to pick up the microRNA from the blood from a tumour-bearing animal, using this technology, this sensor technology that Justin's lab had developed. So it was pretty exciting. So I think the whole nanotechnology space, and in terms of what we can do with sort of nanosensors as well as developing delivery systems, it's quite exciting.
You were talking, we've used COVID as an analogy a couple of times, the mRNA, the PCR, like those are all discoveries that happened years before, and that research and that science has been happening behind the scenes, and then it sort of feels like it bursts onto the scene. Of course, it doesn't just pop out of nowhere. It really feels like you're doing the equivalent thing now that's going to feel like it's popped out of nowhere in a couple of years' time. How many years?
I think we are not far off probably to see the first real RNA and therapeutics for cancer, at least in clinical trials, Maria.
Well, they're already in clinical trials.
That's right, sorry. Yeah.
They're already in clinical trials. We just don't have anything approved yet. But look, I'm very hopeful because I think it was a big milestone to get the first RNA therapeutics, sRNA therapeutic, approved a few years ago in these liver diseases. So once you've got, I guess, a therapeutic approved, a first-in-class, it's what we call first-in-class therapeutic, it's easier for things to then start accelerating.
It's like a proof of concept.
Yeah. It was like proof of concept. So the fact that it got accepted there, then it's much easier to, conduct clinical trials and move forward.
And the experience with the COVID vaccine will probably help that too, because we have learned so much more about this class of therapeutics with the rollout of the vaccine. That will help the people who run the clinical trials on RNA therapeutics probably to optimize to those processes as well.
There's a lot of learnings, I think, from
cross-fertilization.
With all the horrible things with COVID, there's been a lot of learnings.
Yeah. So let's take some questions. Again, Slido, the hashtag is RNA Cancer. For an individual with multiple cancers in their body, and with multiple altered genes, how would this affect the RNA-based treatment?
Well, look, that's actually a really great question. I think often you try and when the genetic sequence of a tumour is looked at, you try and look, you do a hierarchical thing of the sort of genes and ones that you know about, because not all the genetic alterations we identify do we actually know exactly what they're doing. But there are also a large number where we do know. So where you find some more common genetic alterations where it's known what their function is, and you may have 2 or 3 of those, in theory, there might be opportunities there for targeting multiple genetic lesions. First we've got to get the single agent approved in the clinic, and then move towards this multi-targeting.
There's a couple of people asking similar questions along the lines of how do you ensure that the sRNA doesn't affect healthy cells and normal processes?
Yeah, I know that's actually an excellent question, and what we do is that we try and go after cancer genes that are not expressed in normal healthy cells. So
the point of difference.
Yes, that's a point of difference. And often, cancer is an unusual disease. Sometimes you get genes that are driving the growth that are usually part of, say, embryonal development. And during, so when we're embryos, we're, really tiny little things growing, they're required for the normal growth and development of your organs and all the features of you being a baby. But they get switched off at different times when they're not needed, right? And they never get turned on again, as when you're an adult. But sometimes in cancer, you get some of these genes being switched on. And so often they're not expressed in normal cells, and particular genetic mutations are not found in normal cells. So the good thing is, even if the genetic material does end up in normal cells, healthy cells, it's not going to target anything. It'll stay there, it'll get degraded, and it'll get passed through. So the delivery vehicle might deliver it, but there's nothing there for it to target to cause a problem.
A question for you, Palli. This is the most popular question on Slido at the moment. Are there any possibilities for students at the RNA Institute, for example, honours and PhD students?
Absolutely. We are, and you can talk to any of us, not just me and Maria. We are actually a collection of 21 academics from the Faculty of Science, Engineering and Medicine. So you can do engineering, you can do chemistry, biology and medicine and biomedical engineering and
and work across those,
and work
which
across
is what we encourage.
and as I say, Maria and I, we've collaborated now for nearly 10 years, and in fact, by sharing students and their opportunities to do more of that.
Bring your brains.
Come and join us. Yes.
Bring your brains to UNSW. Maria, we were talking about diagnostics before. One cancer that's really hard to diagnose is pancreatic cancer. Mark's asking about that. What's the prospect for early detection of pancreatic cancer using RNA?
Look, there's the biomarkers. It's the same for any type of cancer. Once you identify the appropriate biomarkers that tumour is releasing, then again you can monitor the blood. So it's the same principle. Look, these are all difficult to treat cancers. Like pancreatic is one of the real problem cancers because it's a very complex disease, and it's also very difficult to cure because not many drugs get across. And it would have been great if Josh McCarroll was here tonight. He would have talked about some of the work we all collaborated on with Phoebe Phillips as well on pancreatic cancer, where we were able to show some nanoparticles could deliver sRNA into pancreatic cancer. And that's quite a very difficult thing to do. And the fact that we could look at these, see that they could get into tumours and they could switch off the gene of interest and reduce the growth of the tumour cells was a big thing to achieve. So there's a lot of effort around that, and I think like other cancers, again, monitoring in the blood for markers of that pancreatic cancer, in theory, are feasible.
We'll get back to a few more questions in a tick, but I wanted to ask you both what your sort of personal motivators are to stay in this space. What got you interested in RNA in the first place, and what kind of keeps you going?
What, do you want me to start first, or do you want to start first? You start first.
Yeah, my journey is maybe a bit different. I started from a very fundamental point of view, and I mentioned earlier that I kind of started with my PhD, which has nothing to do with RNA directly, but rather aspects of understanding how life originated, and I got fascinated by RNA as the probably key molecule in that story. 5 years ago then I got to learn about work that biologists like Archer Fox at the University of Western Australia are working on, understanding one of these many myriads of RNAs in the cell called non-long coding RNAs. Turns out they form like gel-like droplets inside the cell. We're still trying to understand what that means. Now, in the meantime, I've been working for the last 10 years in the NanoMedicine Center on peptide-based gels. And again, from a very fundamental point of view, I thought this is really interesting. So RNA and peptide can form these gel things. And I was thinking about it both from a perspective of helping people like Archie and the biologists understanding what does that mean in terms of its function in biology, right? It comes back to the starting point. RNA does much more than just being a messenger. But also from the prebiotic perspective, if you get peptide RNA droplets, maybe that's how the ribosome evolved, and then we got life. That started me off, and then when COVID hit, I sort of realized too. Well, actually, this is maybe now is the right time to start to talk to people here in Australia about why don't we make this locally. And I teamed up with people around the country in the Australian RNA Production Consortium. And we've been working together actually for 2 years of promoting the idea of locally manufacturing RNA, and it's starting to work. So that's sort of my motivation. But I think it would be great if we can cure cancer also with it.
So my motivation is very personal.
Yeah.
I had cancer when I was 21 and went through sort of fairly intense chemotherapy and, ended up in hospital multiple times because of side effects from the treatment. And in the same year, I also started the Children's Cancer Institute. So I got to meet, they were very understanding about all the time I had off, but I also got to meet a lot of children with cancer and families, and parents with kids with cancer. And it really struck me, okay, I'm 21, I sort of understand what's going on. These kids don't, and I could see all the impact for them. And I think that was a real turning point for me to really want to develop a career, because I was doing my undergraduate degree at the time, part-time, and I just thought I want to do a PhD and change things, And unfortunately, when I was doing my PhD, my brother died of pancreatic cancer. So that's a particular cancer I'm particularly interested in. For me, the drivers are very personal. And I think this type of work for me is very personal because I can see the potential to develop less toxic therapies, because I think particularly with children, nowadays 1 in every 900 adults in Australia is a survivor of childhood cancer. And at least 30% of them are going to have long-term side effects from their treatment. So let's do it better. And let's do it with no long-term side effects. That would be fantastic.
Wow, yeah. It's listening to both of you, my personal love of health is this intersection between the wonder of science and the human side of things, and that's really where this unites. Let's take some more questions from a Slido. Someone's asking about personalised medicine and basically wondering whether studies into mRNA and siRNA, whether they're going to lead to the future of personalised medicine as a whole, whether this is maybe like the vanguard for personalised medicine more broadly.
I think it's probably what we said earlier. I think it's going to be part of the package. Do you know what I mean? Like, I think it's going to be, understanding the genome, understanding the targets, and understanding the best way to treat patients. It's all part of it. So I think of it as it's a better, a better package of treatment. And I think there'll be more options off the shelf than we currently have, and hopefully, less toxic options as well. But I think it's really going to help us. Not us, actually. We're doing things to provide tools for the clinicians to treat the patients. And really it's to give them more options for their patients.
Someone really lovely is writing, thank you for the amazing work that you do.
Oh, that's lovely.
Which is nice. The question that I was going to ask, here it is. It's come up to the top because people are voting for it. What are the differences between RNA treatments and CRISPR technology?
Do you want to start, or do you want me to start?
Maybe you start.
All right.
On that one.
So the sRNA treatment we were talking about before, where you can actually target and silence genes in cancer. CRISPR is very different because CRISPR technology, and it's a pretty exciting technology, is really where you have to actually go in and you can change a piece of sequence in the cell. So in other words, but you do have to use what we call genetic constructs to do that at this point. So you have to put them in and you say, well, I want to replace this sequence, or this sequence is missing, I want to put it in, or this sequence is defective, so I want to take that out and put this bit in, which is the right sequence. For cancer therapy, it's probably not something that's probably the high priority, but it is going to be probably more important for gene therapy. So where you have a genetic condition which is not treatable, that's where you want to use that type of technology. But again, it has been used in humans, it's been trialled in humans, but there's a long way to go in terms of doing it. But it's advancing pretty fast.
And it sort of can be delivered in two very different ways. As I say, it can be delivered through what we call a viral vector vehicle. It's sort of how people started with that technology, and I think will still be the main way. But there are ways.
That's where you sort of pop it into a virus that's able to get into the body and not do any.
It's deactivated virus, but it's the way to do it. There is a RNA-based strategy. Where you deliver the enzyme in the form of an mRNA to do the job, and then you need to actually another RNA also to help that system finding the place to do the CRISPR. So it would be very specifically engineered type of an RNA system, but it could work then, and it will probably first be deployed in gene therapies, for sure.
A question which is getting a lot of interest with other listeners, but I think might need a bit of background. The person's asking, could RNA ever encode non-genetic information like has been done with DNA?
That's a little bit of a tricky one. That's got to do with well, I think what they're talking about is how you have coding and non-coding RNA.
Yes.
Yeah, I think, but can that be done with RNA? It's a bit more tricky because you have the DNA and then the RNA sort of.
Well, I think that's a fascinating question because when you asked me what's my personal motivation, I started from a chemistry point of view learning about these non-coding RNAs, and so the biologists are still trying to tease apart exactly what they do now, because they seem to have jobs like regulating gene expression, but some people seem to think they have other jobs as sort of housekeeping in the cell and even structural elements and even metabolism. Now, if that's true, then the next question is, if these things are going wrong, that might be when we see some certain disorders. So then the next question is, how do we fix that? And perhaps one way we could fix those diseases in the future, once we understand the origin of them, is actually through interfering RNAs.
Yeah. Well, yeah,
it's not impossible.
Yeah, because you have these things called long non-coding RNAs, which is probably what they may be referring to as well. And so some of those are known to have functions. Some are not known to have functions. Actually, some don't, some do. But again, people have gone in with sRNA to silence those to see what effect it has on cancer growth. And in some cases, it can suppress cancer growth when you do that. So would we put them in, though? I don't know that we'd put them in, but I don't know. No, we don't know enough about it.
One example
Sounds
of
like you're working up a research question right now as we speak.
No, but one example are called expansive RNAs. I don't know if you like with this CGU repeat, so they get this sort of weird repeat things. Now they interact with proteins in the cell, and there are people now developing actually small molecule therapies then that interfere with that protein RNA interaction to stop a certain disease to develop. But you could also probably go in there with RNA. So yeah, I think it's a good question.
There could be something in there.
We just need more research students to answer all these questions.
Apparently there are places at the RNA Institute. Yeah. Another probably a simpler question to answer is, can RNA be injected directly into the tumour? I think this might be in response to our discussion about delivery before. Yeah. Look,
you can inject it directly in the tumour, but it doesn't, it's very unstable in the body, because we have in our bloodstream, etc., and in our body, we've got a lot of enzymes, and they chew away the RNA, they break it down very quickly. So it can be done, but again, as a cancer therapy, it's probably not the best, because often patients got multiple tumours, particularly the more aggressive cancers have got multiple tumours. So trying to do that with every single tumour is very difficult. So, but it's possible, but it's not very stable unless it's encapsulated in something.
And I think the only way we might see that is if people develop further something which has been known for decades, that you can, with electrical stimulation, sometimes get RNA and DNA, for that matter, directly into a cell. Tricky part is that most of the time when you do that, you kill the cell.
That's less good.
Yeah, but as I say, we were very smart electrical engineers in this country. Maybe one day they will work out the way to stimulate the cells exactly the right way with the little microelectrodes to pump in your RNA.
If we can imagine it, we can create it.
I love that.
It's the magic was the journey, the nano submarine that goes around the incredible journey.
Oh yes, the incredible.
So eyes to the horizon, what does this sphere look like in the next 5 or 10 years, or what's it going to take to bring this sort of tantalising glimmer on the horizon closer more quickly?
Look, I think it's money to do more research. I mean, I know we always talk about money, but I think, we need to do the research. I think we're really at a really exciting stage of development and understanding of these technologies, but we do need the resources to be able to invest in doing this work properly, doing proper clinical trials, do all the safety testing as well. You never want to do any harm, so the important thing is that we know that everything we're doing is for good. Right? I was going to say rather than evil, but it's not for evil. It's but really doing good. So I think I think investing, do more research. So I think things are moving very fast, and I will not be surprised if we have another half a dozen sRNA therapies in the clinic, whether they're going to be for cancer or not within the next 5 years. I'll be surprised if we don't have something coming through.
Palli?
I agree with Maria. I mean, what we need now is further investment, but the good news is that I think COVID brought people together in this space. I think you see that in all the different dimensions, and we've seen that here. We brought together people from different faculties. We have also within this state brought people between different universities. Despite maybe the outside appearance being about state competition, there's actually a lot of collaboration happening between people across Australia. I think another amazing positive story here is the collaboration between people in different fields. Like the
you mentioned before.
the medical people, the engineers, the chemists, the biologists, and so on. And bipartisan, I think we have bipartisan support for this in the Australian landscape at least. So we just need just a little bit more support on the financial side, and we can really deliver.
So just briefly to wind us up, if there's one thing you hope that someone watching this takes away from tonight, what's it going to be, Maria?
If I had to use one word, that I think there's hope.
That's lovely. Palli?
Yeah, and I agree with that. Hope, and let's work together.
Give us all your money to do this research. Look, thank you both so much. And on behalf of the Faculties of Science and Medicine and Health here at the University of New South Wales, including UNSW's RNA Institute, thank you so much to our panellists, Professor Palli Thordarson and Conjoint Professor Maria Kavallaris. And thank you to everyone who's tuned in live to this discussion. We hope it's been interesting. I've certainly found it interesting. We hope it's been entertaining and that you're leaving here with something, having learned something new. If you have a spare minute, please use the survey link on your screen, and a link to today's recording will be emailed in the coming days, so keep an eye out for that. Share it with everyone, friends, family, colleagues, strangers on the street. Spread the good word. We hope you enjoyed the session. Thank you and good night.